Hello everyone, in this video we're going to talk about one type of non-Mendelian genetics called sex-linked trait. So we're going to get started with this picture over here. So it shows you a bunch of different numbers. If you are able to see all the numbers, that means you're not colorblind, and if you can't see all the numbers, that means you're partially colorblind or you're not very sensitive to colors. So the reason why this picture is here is because colorblindness is one of those traits that's located on the sex chromosomes. So as a review, there's a difference between an autosome versus a sex chromosome. In a normal human cell, there should be, or a normal human somatic cell, there should be 23 pairs of chromosomes. Out of those 23 pairs, there are 22 pairs that are autosomes. There is one pair of sex chromosome. And when we arrange them, we arrange the first 22 pairs as the autosomes, and then the last pair as the sex chromosome. So the reason why the sex chromosomes are called sex chromosome is because it determines the biological sex of a person, whether you're a boy or a girl. If you're a boy, that means you have, or if you have an X and Y sex chromosome, that will make you a boy biologically. And if you have X and X for sex chromosome, then that will make you a girl biologically again. So far, out of all the monohybrid and dihybrid crosses that we've talked about, those are all genes that are located on the autosomes. So there's a difference between a sex-linked gene and a non-sex-linked gene. When you look at the sex-linked gene, we're talking about a gene that's specifically located on one of the sex chromosome, either the X or the Y. So as you can see on the picture on the right, the X chromosome is a lot longer than the Y chromosome, so that means it can also hold more genes because it has more space for all the DNA. So when you look at the Y chromosome, it's much shorter, and it only has about 78 genes, which is still a whole lot of information, right? But the X chromosome has about 2,000 genes. So usually when we talk about sex-linked gene, we refer to X-linked. What we really mean is X-linked. There are Y-linked genes, but we talk about it less often, and we don't talk about it in our freshman biology class because the X-linked gene type of sex-linked gene is more common. So a sex-linked gene is usually X-linked, like I just said, and the pattern of inheritance is different. And one other thing to take note of is the male versus female difference of the sex-linked gene. Because male and female has all the same 22 pairs of autosomes, but not the same pair of sex-linked, or the sex chromosomes, the inheritance pattern of sex-linked or X-linked genes are different. For certain disorders or certain traits is more common in males, and there are other ones that are more common in females, and we'll take a look at that. Another word that you need to know about is called a carrier. So Marshy is here for emotional support. Let's see how long he can last. We have a carrier. So a carrier means a heterozygote individual. So when we look at a recessive condition, for example, color blindness, that's a recessive condition. If a female does not have color blindness, but is a carrier of the color blindness gene, that means this female is carrying the gene, right? It can pass on this gene, this color blindness, to the child, even though the female herself doesn't have color blindness, right? So that's a carrier. And the males cannot be a carrier because male is XY, so male can either has X chromosome with the dominant allele of the gene, or a Y chromosome that has or an X chromosome that has the recessive allele of the gene. That's the only X chromosome the male gets, right? Because the other chromosome is a Y chromosome. So male either has a dominant or recessive. There's no in between. There is no heterozygous. But for a female, however, because there are two X chromosomes, there are 3 different variations of a certain gene that's X-linked. So a female can have X big A, right? This is homozygous dominant. So if you see a dominant trait, and it tells you that it's homozygous, you will know the genotype will be X big A. And then we have the carrier situation. This is kind of like heterozygous, where you have X big A, X little a. This individual will still show the dominant trait, but it carries a recessive allele on top of it. And then we have X little a. This is our homozygous recessive, and this will allow the female to show the recessive trait. So an X-linked dominant trait, what that means is this trait itself is a dominant version, right? So if you had an X-linked dominant trait, both this type of female and this type of female will have that kind of trait. So those are more common in female because you have kind of a 2 out of 3 chances of having it, right? However, for a male, we have 2 different types of genotypes only. We have X big AY and we have X little AY, and there is no heterozygous. And X-linked recessive traits are more common in males. So if you had an X little a, for example, colorblindness is an X-linked recessive trait. So that condition is more common in male because a male only gets 1 shot of the X chromosome. You either have it or you don't have it. You don't have the chance of being a carrier. If you get an X little a, you're going to have that trait, right? So that's an X-linked recessive trait. So now we're going to take a look at a very basic Punnett square just for the inheritance of male versus female. So we have mother and father. We have X for mother and XY for father, right? If you fill out the Punnett square as you always would, you put the X, right? You put the letters down, you move the letters across. You're going to end up with 50% chance of a girl and 50% chance of a boy. That's what normally happens, right? 50-50. So when we talk about an X-linked trait, you need to be able to set up this Punnett square exactly the way it is. And then you add the capitalized letter and the lowercase letters on top of it as a superscript. So the inheritance pattern of an X-linked trait, just a little bit more female, has to inherit an X from mother and father, right? If you have 2 X chromosomes, you have to get 1 from mom, get 1 from dad. You can't, if you had an unaffected father, right? If the father is X big A Y, then the female child is going to be X big A, X little a, or X big A, X big A. So in that case, this female is not going to have the condition at all if this condition is X-linked recessive. The male gets an X chromosome from the mother, and the Y chromosome is from father, right? Because the male is XY, and only the father has the Y chromosome and has the ability of passing on a Y chromosome. So once a son gets the Y chromosome from the father, everything on the X chromosome is determined by mother. So whatever trait mother has, whether it's a certain condition or not, is going to be passed on to the son. So your X-linked genes are completely determined by your mother's X chromosome. All right, so now we're going to, there are 2 examples that we're going to talk about. There's hemophilia and then there's colorblindness. Both are X-linked recessive genes. So that means in order for a female to get hemophilia or colorblindness, she has to be homozygous recessive. And for a male to have it, you have to have X little a Y or X little h y. So now we're going to do a practice. Okay. All right, so we have fruit flies. Red eye is dominant over white eye, and we have a male red eye fly and a female white eye fly. So here's how we're going to do it. When you read this question, you always want to figure out the genotypes of the parents first, right? As always. So if we have red eye dominant, how we express it is because you need to make sure to understand that this gene is located on an X chromosome, so you're going to do X big W to represent that. And then if you were looking at white eye, then that's going to be expressed as X little w. So these are the alleles, how you are going to represent the alleles. So when you look at a male that's red eye, you have to have the dominant, all right. So when you look at these genotype, we have male for red eye. That's going to be X big W, and then we're going to have Y, right? That's how you're going to set up the genotype of the male. And because during the Punnett square, we have to separate the 2 alleles of each gene, or 2 alleles for one parent, right? Because during the formation of gametes, during meiosis, we have to separate the 2 alleles. So we separate them on top of each square. And then we have mother, so female is white-eye. So the only way that you can get white-eye female, which is a recessive trait, is X little w, and then X little w again. Now you're just going to fill out the entire Punnett square the way you always do, but remember, you're still going to put the dominant allele in the front, and then the recessive allele after it, right? So now we're going to have the same thing for this box over here, and then this one is going to be X little w, Y, and then X little w, Y again, right? So if you look at the answer key, it's going to show you the same thing. Once you set up this Punnett square, another challenge is to understand which one of these is a male, which one is a female, and which one has the dominant trait and which one has the recessive trait. It's not really a challenge, right? But you need to be able to do that. It's kind of adding on a layer because we're not just looking at a certain trait and whether it's a dominant or recessive trait or the dominant or recessive genotype. We also need to look at whether this is a male or a female, right? So when we look at the questions, the first question on your notes is male to female ratio. The male to female ratio will always be 1-to-1 ratio. That should be 1-to-1. The red eye to white eye ratio. So if you look at these two females over here, because they're heterozygous and they have one dominant allele and one recessive allele, they are both going to be red eyes. And then for these two males, because they only get one shot and the only X chromosome that it has carries the recessive allele X little w, so both of these are going to be white eyes. So our ratio for red eye to white eye is going to be 1-to-1. And then the chance of having a white-eyed male. On the test, I'm going to specify whether I'm looking for a white-eyed male out of all the male or a white-eyed male out of all the children. So in this case, what I mean is white-eyed male out of all the offspring. So both of these offsprings are white-eye and they're male. So that's 2 out of 4 chance or 50% chance. The chance of having a red-eyed male is 0, right? The chance of having a white-eye female is 0 because both of these females that we can get have red eyes. And then the chance of having a red-eye female out of all the children or all the offspring will be 50% as well. So this will be 2 out of 4, right? 2 out of 4 children because each one of these boxes represent 25% chance. So in this example, we have hemophilia. So we have hemophilia is located on the X chromosome as well, and it's an X-linked recessive condition, which means X little h will represent hemophilia. So it's always good during your practice to actually write out what the alleles are and what they represent. So even when I do my questions right now to this day, as your teacher, I still write it out just to make sure that I don't get confused about which one is which, because the normal condition isn't always the dominant, right? Sometimes having a certain condition could be the dominant trait. There could be an X-linked dominant condition, right? So you want to make sure that you're reading the question and you're writing down the notations as you're going along. So when you look at this question, it says a woman who's a carrier for the hemophilia gene. So that means this woman is X big H, X little h. This woman is going to show the normal phenotype because it's heterozygous, right? So the dominant trait, the normal condition, is going to be the actual phenotype of this lady. And then we have a man who is normal, and they want to find out the chance of having a child with hemophilia. So why is this important, right? Sometimes people need to consider, do I want to have this child or not? What are the chances of this child getting a certain genetic disorder? You want to figure out before the child is born sometimes, right? So in this case, the male is normal. So the only way that this male could be normal is X big HY, right? That's the only possibility. We either have X big HY or X little hy, and this male is normal, so it has to be X big HY. Now we put the letters together. We have X big H, X big H again. Here we go. And then we have X big H, X little h. So I just want to show you this process of filling out a Punnett square, so you know exactly what to do when you do your own question. X big H, Y, and then we have X little h, Y. Okay, so now that your Punnett square is filled out, hopefully you filling out this Punnett square was a whole lot less of a struggle compared to me, right? So you can see this is what you have. Now you're going to answer some questions, right? You can look at the Punnett square and ask yourself questions, right? What is this? Is it a male or a female? This is a female. Does it have the condition or not? Is it normal or hemophilia? This is normal. And then this is another female. She's also normal, but she is a carrier of the hemophilia gene. This one is a male. He is normal, and this is a male baby as well. That's hemophilia. So it's important for you to remember that these Punnett square are not actual people, right? They're not actual offspring. This is a prediction of what are the chances of having a specific genotype or phenotype for a particular offspring, or if you were to have a whole bunch of offspring, what are the percentages of a certain offspring having a certain trait? So when we look at the questions, male to female is always 1-to-1. Hemophilic to non-hemophilic would be 3-to-1, right? Because these 3 boxes all are all normal, and then this last one is hemophilic. And then we have the chance of having an affected daughter out of all the children. So on the test, again, I will specify whether I ask you affected daughter out of all the daughter or affected daughter out of all the children. So if I were to ask you chance of having affected daughter out of all the children, well, it's going to be 0% anyway, right? The chance of having affected son, however, so this is an affected son. It will be 1 out of 4 chance, or 25% chance, if it's out of all the children. But if I were to ask you chance of having an affected son out of all the sons, then we're already specifying that we're looking at a son. Then your answer is going to be 50%, right? I'm going to say that one more time. If the question asks you chance of having an affected son out of all the sons, then you're only looking at the sons. Then in this case, it will be 50%. But what I'm really asking in this question is chance of having an affected son out of all the children, then that's 1 out of 4 chance, and that's 25% chance. I hope you learned well from this video, and I hope you could do the practice just fine.